now. For those of you that need to, you should be able to turn on the closed captioning if you want. So I appreciate everyone joining us this morning on the first day of what is apparently our second spring break. I've had the privilege of knowing Twyla since her freshman year. So she was in my Bio 150 class her first semester here. And it was pretty obvious right away this was someone who was very enthusiastic about science and research and would be someone who got heavily involved in the department, even though she did not start off as a bio major, which, I mean, that was sort of a red flag, but whatever. Twyla was probably she was the first freshman I ever allowed to work on the reptile collection. She was also the first freshman that I let become head caretaker. And so she jumped right in and made an impression right away. I think after her freshman year, she was volunteering in 3 separate research labs at the same time, which I don't know if those labs knew it, but Twyla certainly has been ambitious her entire career and really involved in lots of different aspects. She's helped with the pre-vet club. She's helped teach vet tech classes. She's done a lot of outreach and education. She started her own gecko adoption organization and has reached probably hundreds of people through those geckos and those adoptions. So when she changed to her major to biology and had talked about doing the 3-2 accelerated master's program, I was really thrilled to hear about that. And I honestly didn't think she would pick my lab because my lab is small and basically unfunded, and she could have probably gone to any lab she wanted to, but fortunately she picked our lab, and she has really become sort of the anchor in the lab, driving a lot of undergraduate projects, training a lot of undergraduates, and has really spearheaded several changes in direction of my research lab. So we've done, we're with transcriptomics, we've done some immune function now that you'll hear about today. All things that I probably would not have done had it not been Twyla's enthusiasm and drive and really pushing the lab to go in these directions. So I hope she leaves NDSU here thinking that she benefited, but I'll tell you to begin with that my lab is certainly better for having her in it. And I think, we're going to see the benefits of that of years to come as we work to get some of these things published. So without further ado, we'll turn it over to Twyla, and she'll tell us about her thesis research.
Well, thank you, Matt, for that really nice intro. And I am 100% a better and change person because of everything you've done and everything this department and the school has done for me. And I'm so glad I decided last minute to come here. So without further ado, today we're going to be discussing the question, is heat causing erectile dysfunction? On a more serious note, we're really looking at the effects of incubation regime and maternal transgenerational plasticity on performance in the leopard gecko, Evophlarus macularis. So our story starts with 4 individuals. We have Frankfurt in the green collar, Dubai in our blue collar, Juneau in the yellow, and Boston in the red. And these 4 individuals are very similar in a lot of ways. They were all born and raised in Stevens 210. They all came from a subset of our parents who I also raised, and they're all really stinking cute. But there's one thing that makes them all very different that you wouldn't know by just looking at them. And that is that their thermal incubation environment is widely different. So what effects might that have on physiology? Temperature has ubiquitous effects on many aspects of physiology, regardless if you are a reptile, a mammal, an insect, or even plants. It has such strong effects that are only going to be compounded and further edited with the event of global climate change. Specifically, temperature impacts things like enzymatic reaction rates, driving that entire progression of becoming more efficient or less efficient. It affects things like reproductive success. When you become reproductively active from the transition from winter to summer, it has a lot to do with determining the sex of animals in certain species. It also has a lot to do with energy conservation and utilization, as again, these are being driven by enzymatic reaction rates that are determined by temperature. And finally, it affects a ton of biochemistry, specifically looking at metabolism and their rates. One fundamental process that temperature plays a role in is sex determination. In the taxa Reptilia, there are four main types of sex determination. The blue, red, and yellow are all genetic sex determination. But today we're going to be focusing on that green piece, which is around 25% of the reptile taxa, which is our environmental sex determination. ESD has three main forms, and today we're going to be talking about temperature sex determination. To make this even a little bit more complicated, temperature sex determination has three different modes of working, and we're focusing on type 2 TSD. In type 2 TSD, which the leopard gecko follows, that has the majority of males seen on the y-axis being produced only at intermediate temperatures, whereas females are produced both at the low end and at the high end. Now, these temperature thresholds are different among species, but for the leopard gecko, the low end of the spectrum is 29 degrees. That middle area where males are predominantly produced is at 32 degrees, and the high end of that spectrum is at 34, representing kind of our upper critical limit for the leopard gecko. So why should we study temperature sex determination? Even in Reptilia, TSD only makes up a quarter of the sexual methods that are used. And while that is true, by looking at this figure on the right here, we can see that evolutionarily, TSD was extremely dominant. And while it has been lost in clades coming forward, you can see that it has been maintained in more than a few clades just in the gecko family. It has such large evolutionary significance that even if you've made the transition from TSD to GSD, temperature sex determination has its roots. So in the realm of temperature sex determination, which has been a huge scope of literature for a long time, what are the main things that we know here? Of course, the biggest area of research and the longest area of research in these temperature-like studies is understanding the sex determination mechanisms, how this is working and why it's working. Specifically, under that scope of sexual determination, we know a lot about thermal tolerance of embryos, and we know a lot about what high or low incubation temperature does to cellular stress mechanisms in the embryonic stage. We also have a good understanding or beginning of an understanding of how temperature impacts hatchling performance. Most of these studies have been done using a static incubation effect, and most of the taxa include sea turtles, as they're kind of the model animal when we're talking about global climate change, as they've been kind of the poster child for the last 20 years. There also has been some work done on small gecko species and looking at sprints. So these are the kind of two main areas that we know very well in TSD. But what about things we don't? And really, anything outside these two main questions has been ignored. What we're going to be focusing on today are two drastically understudied areas of TSD that are crucial to understanding our climate change scenarios going forward. The first area of focus that we're looking at is trying to mimic natural nest fluctuations by creating what are called fluctuating thermal regimes rather than static thermal regimes. So this figure here is looking at the natural nest fluctuations of the soft-shell turtle. And while this graph looks a little bit crazy, what it's showing you is that over time on the y-axis and temperature, or excuse me, on the x-axis and temperature on the y-axis, this gray line that's bouncing around everywhere is the natural nest fluctuations. And what that's demonstrating is that there are these huge swings in daily cyclic cycles to incubation temperatures in the nest, which under most of the studies so far, that fluctuation aspect has been largely ignored, which is going to make any results from these animals that lay their eggs really close to the surface, such as these turtles and leopard geckos, probably overemphasized, being that they're not truly taking in the natural environment that the eggs are going to be in. Our second main focus is thinking about the idea of how climate change through multiple generations is going to have a significant effect on physiology. The study that I'm going to talk about here is looking at a species of coral reef fish. And what they were interested in was looking at how the effects of changing temperature through generations affect sex ratios. So P1 was represented at a present day temperature. F1 has 3 different treatments, either plus 0 degrees, increase that incubation by 1.5 degrees, and increase that incubation by 3 degrees. That the third generation, or F2, again has 5 treatments here. Has 0 degrees, increased by 1.5 degrees, increased by 3 degrees when you have a medium temperature mom, and then increased by 0 degrees or increased by 3 degrees when you have a high temperature mom. So in the F1 generation, we're seeing really no sex ratio skewing in the plus 0 or the plus 1.5. However, in the plus 3, there's a large skewing towards the male bias. As we move to the third generation is where things become really clear that the maternal influence matters. Again, if we just leave it at present day, no sex ratio skewing happened. If you had a medium temperature mom and you were medium temperature, sex becomes a little bit skewed towards the male bias. If you were then increased by 3 degrees, the sex ratio skewed again towards the male bias like we saw in generation 2. This treatment here is what really demonstrates that how powerful maternal effect is. So if you had a high temperature mom, but you were allowed to incubate back to normal temperatures at plus 0, the sex ratio is still skewed, but in the opposite direction. This demonstrates that the point of recovery can only be so effective up into a certain threshold. And probably unsurprisingly, when you had a high temperature mother and you were incubated at a high temperature, the point of life is gone. That is past that thermocritical limit that is compounded by maternal effect. Which leads us into our experimental questions. Our first focus is the question, does incubation temperature regimes, being either at fluctuating or at static, have an effect on performance? The second level of our questions is trying to understand if maternal plasticity has an effect on performance metrics. And we're going to be looking at these questions by both looking at the hatchling performances and looking at adult metrics. For the majority of studies, we've only been looking at hatching performance or really soon after being hatched, and far less have looked at the effects of post-sexual maturity. We really want to understand the longer-term effects of simulated global climate change on these reptiles. So now we're going to take a look at how we bred our geckos and how we implemented that maternal transgenerational effect. So in RP1, we got them from commercial breeders, and unfortunately, we didn't know the incubation temperature here. But those are our grandparents. The F2s are the parents of our study. And we're particularly interested in a maternal effect. And so we had mothers that were incubated either at a neutral temperature at 32 degrees or at a hot temperature of 34 degrees. And then the paternal, or the father, were always kept at thermoneutral 32 degrees Celsius. Our F3 is where we again see Frankfurt, Dubai, Juneau, and Boston. These are our study samples. And so these were either incubated at a high temperature or low temperature. They were either incubated at 27 degrees or 33 degrees. We're pushing on both ends of the leopard gecko thermal neutral zone by going below that temperature at 27 and above that temperature in 33. Then the eggs were either had a static temperature regime, which we'll call STR, or a fluctuating temperature regime, FTR. In the static regime, which we're staying with Frankfurt and Dubai, the incubation was kept at that mean temperature throughout the entire time with a little bit of fluctuation for day and night. In the fluctuating regimes, this incubation temperature ramped up 2 degrees above their means or 2 degrees below the means. So in the 27 FTR, we have a range of 25 to 29 degrees, and in the 33 FTR, we're having a range of 31 to 35 degrees Celsius. Looking at our performance metrics, we were interested in 3 very classical measurements of performance. We're looking at metabolic rate, which we quantified by carbon dioxide production and oxygen consumption. By locomotion, by looking at limb length to proxy speed. And then finally, immune function by doing an antimicrobial capacity of plasma, which specifically targets complement immunity. Looking at our figure here, here's where our breeding schematic is. And then they were kept individually in their houses for 7 months at a neutral 32 degrees Celsius. During these 7 months, the geckos were fed 10% of their body weight every week and were also weighed weekly. And they had water at limited access the entire time. Once they hit that 7 months is when we did our performance metrics with metabolic rate, locomotion, and the immune function. So I'd like to take a second here just to preview what you're going to be seeing in the results sections. These colors that you're seeing are going to be the same colors that are in the results. So again, we have our 2 main temperatures, 27 degrees or 33 degrees, with the sample sizes listed below. Those then are separated into 4 main groups, either being a fluctuating or static. And then the final level of analysis here is again adding that maternal effect. So in this treatment, this at a 27 degree incubation with a fluctuating regime with a high temperature mother. Here's that same 27 degrees incubation, fluctuating regime with a neutral temperature mother. These are the abbreviations that will continue to show up throughout the presentation. And most of the results is going to be broken down here, and then some of them will expand to those 8 treatments. And here are the final sample sizes when we consider maternal effect. We're going to talk first about the beginning of life and our hatchling success, because if you can't make it out of the embryonic stage is really determinant if you're even going to be affected by climate change, because you may or may not even make it there. So thinking first about hatchling survival. On the x-axis here, we're seeing our 4 incubation treatments. And the y-axis is looking at the number of eggs that were produced here. We can see that there is a strong significance by this chi-square analysis. The black bars here are demonstrating the number of geckos that died, and the yellow bars are looking at the number that survived. Adding the next layer of complexity with adding maternal incubation, we are now seeing those 8 treatments. These are all the 33 degrees, and these are all 27 degrees. And it's clear that in the 27-degree treatments, regardless of maternal effect, they're surviving at a much higher proportion than they survived at that 33 degrees, that high temperature. The difference between the maternal influence was not significant. So we can see that hatchling survival is influenced strongly by incubation temperature, whereas the 33 degrees were surviving far less than the 27 degrees. But the incubation temperature of the mothers did not have a significant effect. Looking next at incubation length. We did an ANOVA analysis. Again, our 4 treatments on the x-axis and the days of incubation on the y. We're seeing here both a significant effect on temperature and in regime. The 33-degree temperatures had a much shorter incubation period than they did at 27 degrees. And this has been well supported in most egg-laying animals that the hotter you are, the faster you will incubate. And our results are staying along with status quo. Interestingly, though, when you compare the fluctuating treatment to the static treatment in the high temperature, there is no difference in that incubation length. However, in the low end of that spectrum, there is a difference. That regime, whether you were fluctuating or static, made you hatch at a faster or slower pace. In that layer of the maternal effect, we're seeing really the same sort of relationship. Our overall ANOVA is significant, but the significance is being driven by the differences between the 33s and the 27s. Again, the 33s are hatching faster than the 27s, but whether you had a high temperature mother or a neutral mother did not affect the length of incubation. So incubation temperatures are affected by incubation length. Temperature regime at that low end at 27 did have a significant effect on incubation length, but maternal incubation did not affect the amount of time spent in that egg. Our last metric for the hatchling success is hatchling weight, as hatchling weight has been strongly correlated to hatchling survival. The figure we're looking at now is an ANCOVA, an analysis of covariance. We're looking at the interaction of treatment, listed here, against hatchling mass, listed here. And the covariant is egg mass. And we seem to have an overall significant model, but this is really being driven between that relationship between egg mass and hatchling mass. As the bigger the eggs got, the heavier the hatchlings get. However, there wasn't strong effects of how this relationship was different between treatments. However, there was a mean effect. This yellow line here, the 33F treatment, has a higher mean weight. You see, this intercept is much higher than the other 3 treatments, and it was significantly different, suggesting that they will always weigh more, but that relationship between egg mass and hatchling mass does not change. So there were treatment mean effects on hatchling weight, with that 33F driving that difference, and maternal incubation did not influence hatchling weight. On from the early stages of life into your later stages, we're going to talk about physiological performance. The first avenue that I was really interested in studying, as I came from this background as a microbiology student, was metabolism, which has been popularly coined the fire of life. So it's been well supported that thermal stress may induce higher metabolic rates. And it has been demonstrated in fish, in crocodiles, and among other ectotherms that metabolic compensation has happened at both high temperatures and at low temperatures. And embryos at a higher versus lower temperature have different means of expending and using energy. Specifically, if you're incubated at a lower temperature, you have to expend more energy. And that makes sense. You're spending a lot more time in that egg, and so you need to sustain yourself for a lot longer. So we quantified metabolic performance was using the Q-Box low-range respirometry package. So we use this machine, and this is our environmental setup where they were kept at a 32 degrees Celsius. Each gecko had their metabolic rates measured for 27 continuous minutes, which ended up being around 720 data points per individual. How we quantified the metabolic rate was looking at carbon dioxide production, which is VCO2, oxygen consumption, VO2, and respirometry quotient, RQ, which is that ratio between how much oxygen you consume to how much carbon dioxide you produce. Looking at our results here, these are two ANCOVAs. We did a log-log transformation on the mass and the macromolecule of interest, being that we know that this relationship isn't linear. And so to fit linear models to this, we have to do these transformations to do this type of analysis. So what you're seeing on both of these graphs is log mass on the x-axis acting as a covariate. And we're looking at the effects of incubation treatment on the log transformation of the macromolecule. In the oxygen consumption, there was a significant treatment difference. In the carbon dioxide production, there's no significant treatment differences. Now, looking a little bit deeper into that oxygen consumption, what's really driving these differences? So now we have transformed our data into mass-specific mass-specific carbon dioxide consumption. This is done by taking the raw values of oxygen consumption and dividing it by per unit gram. Again, we're seeing just raw values here and the incubation temperatures and treatments. We're seeing that there is a significant difference here, and that's being driven by this 33-degree treatment with that fluctuating regime. The fluctuating and static 27s were not significantly different from each other, but the 33 regime mattered here, and temperature mattered, as these were both different than these. Adding now the complexity of that maternal incubation, we are seeing an overall significant ANOVA probability value. However, when we are looking at individual comparisons between hot and neutral mother, we're not really seeing that breakdown. Essentially focusing on the difference between 33 fluctuating with a hot mother versus 33 fluctuating with a neutral temperature mother. This does have a p-value difference of 0.05, which is fairly small, but for the ANOVA, it's not a significant value. So what we can take away from this is that oxygen production or consumption is significantly different based upon incubation temperatures. So, and the magnitude of this is influenced by that maternal incubation, where carbon dioxide production was not influenced by incubation treatment nor maternal incubation, and respirometry quotient was not influenced by incubation treatment nor maternal incubation. Our second area of interest was looking at the locomotive performance of our geckos. We know that locomotion performance is really important to early survival, especially as a tiny little gecko that looks like a snack to 20 different animals in its environment. If you cannot escape predation events, you can't move on to be fit in your life. Specifically, it's been demonstrated that longer limbs are correlated to faster speeds. And those faster speeds have a higher proportion of survival. These early thermal environments has been shown to affect anoles, sea turtles, and skinks in a variety of ways. It's not consistent how these incubation temperatures affect these performance metrics, but it's clear that it is important. And these little guys can move extremely fast. For reference, this gecko has just hatched a day ago. And he took off. So we know that they can run fast if they choose to. Knowing this, we built our product design around having our leopard geckos perform in a sprint trial and in an endurance trial. So we built a break-green sprint track to calculate maximum velocity and maximum acceleration. And then we built a motorized treadmill to look at how long can these leopard geckos run. However, we'll show you our first attempts in action. So you can see the gecko is kind of walking on the treadmill here, but it's not extremely consistent. And as far as the sprint track, they could care less what I'm doing to them. They will not run. So we tried a variety of different methods to try and encourage them to run down the sprint track, but they were simply uncooperative. So we had to then proxy locomotion. As we mentioned before, there is a strong correlation between limb length to sprint speed. And so we measured a variety of limb lengths to proxy how fast you'd be able to sprint and how long. So measurements we took for snout to vent length, what we call SVL. We took humerus length, ulnar radius length, femur length, tib fib length, and specifically we're interested in right hind foot, as this length has been strongly correlated to faster sprint speeds in the leopard gecko. How we analyzed this data was by using a principal component analysis. And I want to just talk briefly about this type of analysis, as it's not one that we see or use every day. So a principal component analysis is really a measure of variance across multiple variables, and it tries to reduce the dimensions that we're looking at. So we can take very complicated data sets that are really intertwined with each other and try to make them simpler and easy to understand by reducing dimensionality. So we use the PCA scores to get a score on every individual's metric. So for example, Frankfurt will have a score for femur length, tib fib length, ulnar radius length, right hind foot, and humerus. And they'll get a score for each one of these components. The PCA scores of each treatment are then averaged. And then we performed an ANCOVA. In this ANCOVA, we used PCA scores as a response against treatment with snout to vent length, SVL, as a covariate. We know that snout to vent length and limb length are extremely correlated. Hence, we didn't include it in the principal component analysis, but rather use it as a covariate. There's Frankfurt. The outputs of our PCA are demonstrated in this figure here. So in principal component analyses, the higher up principal or component you're in accounts for the most amount of variation. So principal component 1 has 47% of the variation here. Principal component 2 has far less at 19%. Principal component 3 accounts for 15% of the variation, and that decreases as we go down the order of components. We analyzed in the ANCOVA the first 3 components, meaning that this is accounting for 82% of the overall variation. When we did our ANCOVA analyses, for PCA1, we are seeing no differences in treatment in relation to the average PCA scores. So that's accounting for 47% of the variation. In PCA2, we again are not seeing treatment differences in relation to those scores. PCA3 is where it starts to get interesting, as we are seeing a significant difference between treatment and PCA scores. But remember, PCA3 is only accounting for 15% of the overall variation. However, let's take a closer look at PCA3. So we did an ANOVA analysis on the PCA scores by treatment with dropping snout to vent length, and we didn't see a significant difference here. We did see, though, that the temperature groups were on opposite sides of the PCA component. That 27 degrees were on the negative side of PCA3, and the 33s were on the positive side of that. So what does this mean? We look at the breakdown of the vectors of our components. Here's principle 1, 2, and 3. We can see that the majority of variation, 82% of variation in principal component 3, is related to the femur. So when the 27 degrees are on the negative side of this means that the longer geckos have shorter femur lengths. Being then on the positive side of this means that the longer geckos, when incubated at 33, have longer femur lengths. However, we did look at these mean values, right, with the ANOVA, and there is no difference. It is important to recognize that the range of our covariate is fairly small, with that snout to vent length only varying by 15 millimeters across our entire sample. Furthermore, when we drop SVL and just look at the mean values, there is no significant difference. So while this may be interesting, and certainly something to look at, I think we need a larger sample size to really piece apart what's happening here. The final aspect of locomotion performance is looking at that right hind foot length. We're looking at another ANCOVA that has the same model aspects as before, except that right hind foot length is on the y-axis in millimeters. And we are seeing a difference overall in the model. However, this is being driven by that relationship of snout to vent length to right hind foot length. And there is no significant differences in that relationship by treatment. It is important to notice that the 27 degrees and the fluctuating regime seem to get smaller in that right hind foot length as they get larger, where the other 3 treatments, the relationship is flipped. Again, we might be able to piece this out better with larger sample sizes here. Well, we can conclude that PCA3 scores are significant by treatment, but again, only accounts for 15% of that total variation. And right hind foot length is not significantly different between those treatments. Our final aspect of performance is looking at immunological performance. We have, our 2 classical aspects of immunity. You have our humoral immunity, which is that immediate response, and we have our cellular immunity, which is our memory, and it takes longer to activate. However, we often forget about what binds these 2 things together, which is our complement immune system, which is driven by the immunoglobulins in our plasma. And that's what we're focusing on today for our immunology, our immune tests. So environmental stress is one of the most significant factors affecting performance and health in any taxa. Things like habitat loss, water, increase in precipitation, and of course, temperature will have major effects on our performances here. Really, there's been a very, very limited amount of research done on immune activation in reptiles. Again, most of this research that has been done in turtles. They demonstrated in a species of softshell turtle that higher incubation temperatures resulted in a decrease of immunoglobulins IgM, IgZ, and cytokine CD3. It is also shown in amphibians that higher ambient temperature, so the environmental temperature they are in, reduced bacterial killing ability in bullfrogs. So temperature has been shown to affect immune function, just not very often in reptiles. How we quantified our immune function was with an antimicrobial capacity of plasma, or an ACP assay. How we did this is we drew a whole blood sample from our geckos and isolated the plasma, inoculated the plasma with E. coli that was at 2 × 10⁵ strain, and put that into solution. We then allowed that to be plated and incubated for 2 hours at 27 degrees, and then we read these for their optical density of each sample. The lower optical density each sample had, the more amount was killed by the plasma. And here's our ACP calculation, where it's 1 minus the optical density of the sample divided by the optical density of the positive control. This is the method that was used in that amphibian bullfrog paper and a prior amphibian paper. And so we know that this method is valid for quantifying immune function. So what we see in terms of antimicrobial capacity of plasma is, first, by treatment, there's no significant difference, and we're seeing very large variation in killing ability in all 4 of those treatments. And then with the added complexity of maternal influence, again, no significant difference. We can note that on average, the higher temperatures are killing more. However, this again wasn't a significant difference, and the ranges of each of these treatments is very large. So we can conclude that we know that antimicrobial capacity ranged from 10% killing ability to 70% killing ability. But this was not different across treatments. Although, as I just mentioned, higher temperatures trended towards higher percentage of kill. But again, this was not a significant difference. Something that may be able to tease apart better if we have larger sample sizes. But it's still an interesting metric that we did and we showed and should be done in the future. So kind of recapping all those things we just talked about. How are our samples, how is Frankfurt different from Dubai, then from Juneau and Boston, along with the 34 other geckos that are included here? So first, starting off with hatchling survival. The 33 degrees survived a significantly less than the 27 degrees. In incubation length, as supported by literature past, the hotter incubation temperatures had shorter incubation time periods. With hatchling weight, this, while small, the ones that were incubated at higher temperatures had a lower weight. And that's a reasonable outcome. As you're spending less time in that egg, you're having less time to absorb the different, yolk supply and other things that are provided to you in the egg. Switching over to our performance metrics, first looking at metabolic rate with carbon dioxide production. This is slightly greater in the high temperature samples, but is not a significant difference. Looking at the consumption of oxygen, we're seeing that the high temperatures have they produce oxygen more, or consume oxygen more. Specifically in Juneau and the 33Fs, they have a much higher rate of consuming that oxygen. Thinking about our respiratory quotient, these are nearly identical across treatments. So that ratio between how much oxygen they are consuming and carbon dioxide they're producing is not different among treatments. Moving on to locomotion, specifically in our right hind foot, we're not seeing any treatment differences here. And finally, our immune function procedure using antimicrobial capacity plasma, we're seeing slightly more killing in that high temperature samples, but again, this is not a significant difference. Just give you a second to look that over. Considering our performance metrics and our studies, there's always more that you can do, and there's always biological things that are influencing your study that you didn't intentionally put in there. So talking about our results first with metabolic rate, it's important to note that these were only 30 minutes of data collection. And while this is a good snapshot of what is happening, it's fairly limited to only that short amount of time. We did want to have a much longer time of running this, but we were really restricted by our machine. Additionally, it's important to note the time of day that our metabolic measurements were taken and what time of year they were taken. So we took the measurements between the hours of 6 p.m. to 8 p.m. So a relatively small window, but this is that transition from late afternoon to early evening and kind of getting toward the 8 o'clock time, transitioning to dusk, which is where leopard geckos are more active during the day. So you could be seeing an upregulation of metabolic rate. However, this was taken during the months of November and February, which is a really whole organismal downregulation. Things that we'd like to see done in the future here is first by increasing the amount of time that you take these measurements, by really doing a 24-hour or longer measurement. This will really capture not only daily circadian rhythm of the whole animal, but metabolic systems also have cycles to them. And by doing it over that large range of time, you get a better idea of how they're functioning throughout the day. And it'd also be interesting to take these measurements during an active season, such as during the breeding season in the spring, or just where there's generally more active time. In locomotion performance, we must address the obvious. The geckos did not actually perform. We tried a number of things to get them to do so, but it didn't happen. We were able to still make good predictions of how they might perform based upon their limb length that's been supported in a lot of literature. Something that also may have had an influence on the size is that we really limited our genetic variation of our appearance. All of the mothers were half siblings and all of the fathers were full siblings. And we purposely did this to try to avoid any excess noise in our data. However, the parents themselves did not vary a lot in size. And so this could potentially have an influence on those measurements we took. Something that we'd like to pursue in the future is, first and foremost, find a way to get leopard geckos to run. Our leopard geckos are really calm. They've been around me for their entire life, so they're probably not very scared of me. It's also very noisy in the lab, and so the things we tried just didn't scare them. So finding a way to get them to actually participate would be important. But more interestingly, taking further effects into account would be important. It was demonstrated that 3D kinematics, specifically of the foot posture, makes really big differences for sprint speed. And they show with different incubation temperatures that transition from plantigrade, having your foot flat on the ground, to digitigrade, moving towards that toe running stance, varied when you varied incubation temperature in an anole species. So finding out where that point of transition could be really important and really interesting. Finally, in terms of locomotion, we didn't take into account tail length, tail width, or tail weight. In many species of reptiles, specifically the leopard gecko, their tail is extremely mobile and is really used sometimes as a fifth limb to help with balancing, to help use it as a steering mechanism, among other things. It's been shown that by restricting tail movement will affect how kinematics works in the lower limbs. So that could be something interesting to take into account next time. And finally, with our immune function, we did test only one microbe that is naturally occurring in the gut. So presumably there are some microbial capacity against E. coli, but there probably at the same time is already a natural resistance to it because it is occurring. What we would then suggest is include more bacteria. Specifically, we're interested in Salmonella and a staph, as these were also used in the previous reptile literature. And then we can better understand how these are, how their complement activation is happening in reptiles by targeting complement-specific enzymes to better understand which pathway complement immunity is taking. So overall, we can see that our 33 degrees with a fluctuating temperature is having a lot of interesting effects going on. It's indisputable that temperature is one of the most important characteristics in all of life history, and that's within ectotherms and beyond ectotherms. And when our environmental conditions change very rapidly, even from the day to day, as we can see with a major blizzard in April, there are going to be a variety of consequences happening. So what is next overall for this project? The first thing would be to increase that number of 27-degree animals included in our study. We did stop breeding 27s fairly early because they were taking so long to incubate that we would not get them to grow up to be 7 months old to do our performances in a timely manner. Next, we'd like to consider not only incubation temperatures, but then the environmental temperature, is seeing how instead of keeping them at 32 degrees, have a range of temperatures from high, medium to low to see what the effects of your environment have on performance, which is really gets at the idea of global warming from the day to day. We're also interested in looking at the mitochondrial genome. In a past project we worked on, we have the transcript of the leopard gecko, and we're seeing some interesting things here. But then by specifically targeting the mitochondrial genome, we can potentially get a better idea of how maternal effects are impacting performance, as the mitochondrial genome is inherited from your mother alone. Finally, we'd like to expand this to just beyond the leopard gecko. We propose having 2 species of temperature sex-determined geckos, and then 2 more species of genetic sex-determined geckos, to see if that determination system has an impact overall. So this is an extremely exciting, extremely new phase of research that can be participated in many avenues, and I'm so excited to see where this goes. And with that, there are so many people that I'd like to thank. I couldn't have got here without my committee, without the love of people from my department, and of course my friends, my family, and the 37 geckos that made this all possible today. So thank you for letting me enjoy my time here at NDSU for the last 5 years, and the floor is open for questions.
All right, round of applause for Twyla. And then if you have any questions, go ahead and raise your hand or type them in the chat.
Woo! Yeah, Wendy, go ahead.
She's clapping.
Oh,
Well, but I also wanted to say that that was amazing, and I wish you the best of luck. Thank you so much.
oh my goodness, thank you so much. That means a lot. Yeah, Craig, go ahead.
First off, really nice job, Twyla. That excellent, and
Thank you.
boy, a lot of work. You've done a lot.
So many things.
Given that you saw the pretty dramatic effect of temperature on survivorship and hatching mass, did they catch up in mass by the time you tested them, and were you surprised that you didn't see some metabolic effects in terms of treatments?
Yeah. So to answer your first question, did they catch up? We did analyze 7th-month weight by using an ANOVA of a treatment against weight, and there wasn't a significant difference any longer. However, the 33 degrees in the fluctuating still had a lower overall body weight. It wasn't significant, but it still was lower. So they are catching up, but there are still some differences seen here. to kind of talk about the second part of being surprised about the metabolic stuff, I was very surprised. However, it was supported in previous literature that oxygen consumption alone was different in the event of different incubation temperatures and less strong support for that carbon dioxide production. And so I was surprised, but what we saw was supported by the literature.
Thanks. I have one follow-up question. I noticed on some of your ANCOVA plots all the way through that it seemed like one of the treatments had the opposite slope of the others. And I wondered if you could either bring that back, or is that a biologically interesting outcome, or you know what I'm talking about?
Yeah. Let's see here. Let me reshare my screen. I'll go to student presentation mode. Can you guys see the screen still?
Yes.
Okay. Yeah, so let's find one of these here. Yeah, so like simply looking at just oxygen, for example, we're seeing that 33 degrees having a really almost not opposite and also, but really different interaction. And the biological significance here is that 33F, which is where we're seeing the deviations most of the time, is approaching that thermocritical limit. This is going past what leopard geckos can normally survive. And so it's showing that there are limitations to that upper limit, and it will have negative effect, not negative, I guess, but different effects of physiology as we're moving past what's normally allowed.
Okay. Yeah, I just noticed that 27 was going the opposite direction, but I, after seeing it more closely, yeah. So, thanks, Twyla. Very nice.
Yeah, thank you.
Okay, Twyla, there's a question from Terry in the chat that says, there are so many implications to this work. What questions are you most interested in pursuing or seeing pursued?
That's a great question. Thank you for coming in. I can't call you Terry. This is Egan to me. So some of the most exciting avenues of this research, I think, really are in that maternal aspect. This really hasn't been done in a lot of, in most TSD research. And, you know, even though we're not seeing really significant differences, we are seeing a lot of interesting trends that are happening here. And I think this is somewhere that really deserves a lot more attention. As we showed right away in that fish study, maternal influence has profound effects and hasn't really been considered. And so that's the thing I'm really most interested in. And going along with that, I really think looking at the mitochondrial genome would really benefit in understanding the maternal effects and give us a clearer answer to if or what is even going on here. From my knowledge, the leopard gecko transcriptome hasn't even been published. We're still working on that. And so even less than the mitochondrial genome has been far less explored and deserves some attention, if that answers that question.
You have another one from Sarah M. Do you want to read it, or do you want me to read it for you?
Yeah, I can pull it up. So Sarah, thank you. Oh, you have your class listed. That's so nice. Student questioning, what are the next steps in the research different reptiles looking at amphibian outcomes such as frogs? Oh, that's fun. Note that 180. Yeah, and so anurans are also a TSD species, and they typically follow TSD1, where there's a high proportion of males being produced at a high temperature. So for your class, it might have been that in that laboratory setting or wherever you got these animals from, that they were incubated at more high temperatures than low temperatures. And for the future research stuff, in addition to the mitochondrial performance, I really would like to see how locomotion would be impacted as well. specifically looking at the kinematics of foot transition would be something really interesting. I'm really interested in orthopedic type of medicine, and so this is where I'm naturally drawn to, is kind of that variation in that limb type stuff and how they might be different. Yeah, Ethan.
Notice in your one of those Angola plots you have outliers. How would you keep the outliers?
Yeah. So if you couldn't hear that, Ethan said he's noticing that there's outliers in these ANOVA plots by, these dots are representing outliers and why I kept them in. And I think that's something really important to talk about. There's the simple answer, there's no biological reason to why those outliers should be excluded. There's nothing that happened with them in the methods or nothing that happened with them physiologically that would tell us that something, mechanistically went wrong. So I'm a very big proponent of not over scrubbing your data to make the math look better, because biology doesn't follow the rules of math, and that's okay. We don't want to artificially take out these samples just to make our point better, because there's no reason to take these out. That answer your question? Yeah, Harry.
In this changing world, when looking at organisms with TSD, do you expect that in the near future, will you see less and less animals using TSD, maybe transition to genetic sex determination? Or do you expect that change to be so radical that the frequency will be?
That's a great question. So to kind of just reiterate for the people on Zoom, he asked, with this changing environment, do you expect more or less organisms to use TSD, or what will happen? So I think there are two kind of ways that, or two outcomes to this. I don't know if the proponent of TSD will become more prominent. However, it is interesting to note that in genetically sex-determined reptiles, temperature is strong enough to reverse the sex. There's been many studies that have shown that in GSD, in the proponent of extremely high temperatures, what is genetically male is being phenotypically female, and vice versa. So I think that's a major consequence of this increase in temperature, both on TSD and GSD specifically. To kind of talk about that second part of what will happen to TSD, that's something we're actually looking in our lab. We have a multi-generational study of looking at what happens with cumulative incubation temperature. And there are two kind of ways this could happen again. So we could either see that the sex ratio curve shifts over. As it increases in temperature, the physiology behind sex determination ranges are bumping up. That would be the most ideal situation, in that sex ratios would be still held constant or at least even. Another outcome, however, is that these ratios are not switching. And so as we're moving from, 32 degrees to 34 degrees, we might end up with an extremely female biased populations. And that would obviously mean huge things for the populations in the long term. There's a lot of active research seeing what will happen and how these ratios are changing, but that would be my hypothesis. Does that answer everything?
Yep.
Perfect. Thank you.
You got one more in the chat, and then we're probably going to cut it off here just to be mindful of everybody's time.
All right. From my sister. It was such a joy to watch. You notice any noticeable changes in behavior, overall disposition, and how? Oh, okay. Yeah. So, behavior wasn't, the point of my study, but it's been well documented that the higher temperatures, specifically in females, create really aggressive females. And this is likely because those females are having a higher unnatural level of testosterone for a female to have. And we do know that testosterone is typically associated with aggression behaviors. And so that's what we're seeing in terms of temperature. On kind of another note, for whatever reason, all my geckos were really mean this year. I think I got bit more times this year than I have in the last 4. So something else might be going on, but in terms of research, we do know that higher temperatures typically create more aggressive females. If that answers that question good enough.
Okay, well, join me one last time in thanking Twyla. Nice job. Thank you everyone for coming out. Stay safe and have a great second spring break. I guess we'll see everybody next week. And then the committee, if you would just stick around after everybody leaves and we can make a plan for the private portion of this defense.
Thank you guys for coming so much and stay safe out there.